Our first speaker for this afternoon is Kornelia Polyak, who trained with Joan Massaguet and Bert Vogelstein, and who has been at the Dana-Farber for a number of years, and who will describe the great diversity one finds within tumors and how the existence of this diversity complicates our understandings of the pathogenetic mechanisms.
Okay, so first I'd like to thank the organizers, Bob and Richard, for inviting me and be among this group of very good speakers. And I try to match up both in terms of the quality of the talk, and hopefully some of the pictures I have will also be as colorful and nice as the previous ones have shown. So I'd like to start with a quote from Charles Darwin, which basically says that it's not the strongest of the species nor the most intelligent, but the one most responsive to change that survives. And the reason is because we approaching cancer, breast cancer in my lab, basically using the same kind of principles as Darwin and many of the other naturalists used in studying nature. And basically the reason is that a tumor is basically an ecosystem that harbors cancer cell communities, not individual cancer cells, but communities that live in a physiologically and topologically distinct niches. And just like in nature, similarly in tumors, only populations of cancer cells can evolve. A single cell is not evolving. Only populations of cancer cells can evolve. And replication and cell turnover is very important for the evolution of the tumor, just like diversity and selection are the key determinants of the progression. And this oversimplified view of breast tumor progression illustrates that the tumors go through defined stages, and actually we think that these stages are less and less defined in terms that they're not necessarily occurring in a sequential manner, but actually early dissemination can occur at fairly early stage, but it doesn't always necessarily lead to clinically observable metastatic disease. And breast tumors are very heterogeneous. Patients different tumors in different patients have very different phenotypes. This classification study by Chuck Peru, published many years ago, have identified major subtypes, which they call luminal A, B, HER2 basal-like, and normal breast-like. Since this study was published, more extensive studies actually have shown that even this is an oversimplification of the heterogeneity among tumors, because even these subgroups are not really homogeneously behaving. The subtypes also have very different risk factors for the initiation of the tumor, different response to treatment, and different risk of progression. And even when they progress to metastatic disease, the site of metastasis tends to be different for luminal and basal and HER2 positive tumors. And the question is what is the underlying mechanism that could explain this high degree of intertumoral heterogeneity? And two potential explanations are plotted on this slide, which are not mutually exclusive. In fact, most likely the combination of the two is what's happening. One of them, that maybe the subtypes are due to the fact that the different tumors initiate from different normal cell, such as a basal-like breast cancer, which very much looks like a stem cell, may initiate from this bipotential mammary epithelial stem cell because it has the features of those cells. And the luminal cancer cell, which is more differentiated, may initiate from a luminal progenitor. Alternatively, we can also imagine that you have the same type of progenitor that can generate multiple different tumors because of the genetic and epigenetic events that occur in these tumor cells. And as I mentioned, most likely the combination of the two is what's really happening, because we know that the same change in not causing the same effect in two different normal cells. In addition to this intertumoral heterogeneity, there is a very high degree of intratumoral heterogeneity, meaning even the same tumor of the same patient at every given time is not homogeneous. This study published by Craig Allred last year have shown that even this early stage tumor, which is ductal carcinoma in situ, just looking at three markers such as the ER hormone receptor, estrogen receptor, ERB2, and p53, you look at the staining pattern in a subset of tumors, you see very highly heterogeneous staining, such as completely negative and positive area within the same tumor. And in the case of HER2 and P53, this may actually reflect genetic heterogeneity, although it was not studied in this paper, because amplification of HER2 is commonly associated with the staining, and also P53 mutation is commonly associated with positivity of P53. And the question is, like, what is the underlying mechanism that can explain this high degree of diversity? Because we know that with very rare exceptions, tumors do originate from a single transformed cell. And Sean already kind of alluded for these different models that have been proposed to explain this heterogeneity. I just go over them briefly. So basically, one model initially described very nicely by Novel in the 70s have proposed this clonal evolution model, which is very similar to the Darwinian selection in nature. Basically, it says that there is a genetic instability of the property of the tumor cells, and this genetic instability combined with a clonal selection is what drives tumor progression. This model, again, recently has been referred to as the stochastic model, but as Sean also stated, this model doesn't state equality for properties. In fact, it emphasizes heterogeneity for properties, including self-renewal and tumorigenicity. Then another kind of hypothesis that was proposed that explains intratumoral heterogeneity is this malignant or transformed stem cell model. Basically suggests that maybe some tumors initiate from a normal stem cell, and in certain cases that stem cell can maintain some differentiation potential, which in the case of teratomas could be almost full normal differentiation at some tumors. But in other tumors, it's maybe just some kind of aberrant differentiation, not really normal differentiation, just kind of switches in phenotypes. And then lastly, Bob Kerbel kind of accidentally discovered that epigenetic diversity can actually lead to much higher degree of diversity in the tumors than genetic changes alone. And basically, they found that metastatic ability of cancer cells, like population of cancer cells, frequently can be explained by epigenetic diversity within that population. So as you see the dates of these papers, and again, these are not the earliest papers that propose these models, these ideas have been around a long time, and so basically this kind of diversity issue is not really new. We just have better tools nowadays to study this diversity. So my lab, we have been looking at breast tumors at each level, basically assessing genetic heterogeneity, epigenetic heterogeneity, and also looking at the phenotype of the cells in terms of stem cell and more differentiated markers. And what I will show you today is that basically we see heterogeneity at every level, and these seems to be overlay on top of each other, and we think each one of them is important, and the relative importance of each mechanism could be specific for the tumor and to the stage of the progression. One of the difficulties that we have in a mammary gland, particularly in a human mammary gland, is that we don't actually have a good definition of the hierarchy of the normal mammary stem cell differentiation, and we don't even have very good markers that are uniformly can be used to identify the cells. This is a hypothetical, tree proposing that there is a bipotential mammary epithelial stem cell which is thought to be negative for hormone receptors and HER2 and positive for some markers, some mesenchymal markers. And then there are two main lineages, the myoepithelial cell lineage, which forms the outer layer of the ducts, and then alveoli. And also there is a luminal epithelial lineage, which by itself is still further heterogeneous in terms of there are ER negative, hormone receptor negative, positive cells. And also there is a ductal epithelium, which is likely to be very different from the secretory epithelium that produces the milk during lactation. But as this question mark illustrates, basically we have very rudimentary knowledge about these different stages and what are the markers that identify these cells. And this study that now has been published almost 6 years ago by Mark Rugg's group have identified some of the markers. These are some studies that study the normal mammary epithelial stem cells, and various markers have been proposed by different groups. And these are just a list of the various markers that have been proposed. And as you see, there is not really a good, consistent agreement in terms of what are the markers of the putative progenitors and the stem cells. And as I mentioned, like the Mike Clark's group published now almost 6 years ago this study, which created lots of interest and in this area. In terms of what they found is that the CD44 positive, CD24 negative, lineage negative breast cancer cells seems to be more tumorigenic when injected into the fat pad of NOD skid mice than the CD24 plus cells. And based on this assay, they determined, or they stated, that these cells, the CD44 plus, 24 minus cells, seem to be like these breast tumor initiating cells or breast cancer stem cells. And based on Sean's talk, actually you can tell that these assays have been since then criticized, and in terms of definition of a cell cannot depend on an assay, that depending on how you do the assay, you get different results, since if you're using different strain of mice and wait different time, different way of injection, you get different definitions of the cells. One additional point I'd like to make, that these single cell assays are very artificial, because if you remember, tumors in humans take years, if not decades, to evolve. So basically, a single cancer cell taken from an advanced stage tumor is not expected to reproduce the whole tumor, because it was not evolved to be independent and growing on its own. Maybe the very early initiating cell could do that, but that's usually not what people study. So instead of using many of the assays, my lab has been mostly focusing on trying to understand the molecular heterogeneity of breast tumors and correlate this heterogeneity that we see with clinical responses in patients. And the approach we have been using over the years is two different ways. One of them is to dissociate the tumors and normal tissue into single cells, and then capture the various cell types using cell surface markers that are specific for them, such as capturing the CD24 positive cells and the lineage negative CD44 cells as kind of representative examples of the more differentiated and less differentiated cells. We have done this study from normal tissue and also for multiple tumors, and this left-hand side shows this comprehensive gene expression profiling of the cells from different tumors. Basically, the conclusion of the gene expression profile is that indeed the CD44 plus cells seems to be expressing many markers that are characteristics of stem cells, and they also enriched for genes that involved in invasion angiogenesis and cell proliferation, as shown on this gene ontology enrichment side. So basically, that's consistent with the idea that these cells are more different, more stem cell looking, and they're more likely to be responsible for potential progression of the tumor. But then what we noticed as we started to get into these heterogeneity in more detail, that actually if we're looking at tissue sections of tumors, and for example, staining for these markers CD24-44, or even if you're combining them with other markers like ALDH or any other that you want, if you look at different area of the same tumor, so this is a microscopic image of the tumor, and then 4 areas I highlighted here, we look at the staining pattern, it's very different. In some areas, the 2 cell types seems to be almost equal frequency. In other areas, the CD44 plus cells seem to predominate. So then the question is how can you interpret this kind of diversity? And is there any particular pattern within the tumors that could explain where the different cells locate? In addition to seeing this diversity within the tumor, we also see differences among the subtypes of breast cancer, such as I mentioned that the basal-like breast cancer is actually almost all the cells are positive for CD44, which is this more stem cell-looking marker. And in the luminal tumors, we see almost all of them positive for CD24. So basically, these markers fluctuate within the tumor and also among the tumor, and the frequency of the cells varies, from few percent to virtually 100% of the cells. But then one of the major differences that differentiates cancer from normal and normal stem cells in particular is that cancer is genetically abnormal cells. And in order to study the tumor, we cannot forget about looking at the genetic alterations in the tumor cells. And particularly in this study, what we wanted to see is the CD44 plus and 24 plus cells isolated from the same tumor of the same patient at the same stage. Are these genetically identical or not? Because the prediction of the cancer stem cell model would be that they have to be genetically identical if they are direct progeny from one another. So we assess this in two different ways. One of them is looking at SNP arrays, basically using the same population of cells that we ran on expression profiling. We also ran on SNP arrays. And as you see, the different populations from the same tumor overall seem to be fairly similar to each other, even not almost identical. But then when we decided to look at single cell level, which gives you a much higher resolution of heterogeneity, in this particular example, we did a metaphase spread of a primary cultured cell and then hybridized for a chromosomal region for 8q24, which is frequently gained in breast cancer. And then what we found that each, both the CD24 plus, 44 plus population had multiple copies. But then for another region on 1q21, the CD24 plus cells had multiple copies, like 4 or 5 per cell, and the 44 plus cells were completely normal. So what this shows that in this particular tumor, the two cell populations are genetically divergent. At some point of progression, even though they seem to come from one clone, they evolve differently, and they are not the direct descendants of one another. More recently, we went even further and have done these immunofish studies in a larger set of tumors on tissue sections. And this is an example of comparing DCIS to invasive areas. This particular case is a HER2 positive tumor. As you see on an immunostaining for HER2, even HER2 staining is not homogeneous within a tumor, as I mentioned earlier. The CD24-44 staining, again, is not homogeneous. And I don't know how well you see, but basically this HER2 copy number is not homogeneous. If you start counting single cells, it's actually a very high degree of heterogeneity that we can see. Then in this particular DCIS, we actually saw a very nice example of actual evolution of a subset of tumor cells, because as you see, like the majority of tumor cells seem to have normal copy for this particular chromosomal region, and the subset of them seems to have amplification. And in the invasive area of the same tumor, this clone seems to predominate, and the other cell became a minority. And we have been looking at this at different stages of progression using this immunofish approach. This is looking at a metastatic sites of the same patient, again, the same patient, multiple different sites of metastasis. As you see, like yellow color corresponds for CD24 staining, blue color for CD44. Just comparing the lung and a spleen and then counting again the copy numbers for this particular locus, these metastases are not the same. They clonally and genetically very divergent. And as you see from the colors also, even the phenotype of the cells is very different. And again, this is another example on another patient. Similarly, normal copy number in the lung for this particular locus, amplification in the liver, and also the CD24-44 population is different in this case as well. And then we wanted to see, like, how can we actually quantitate this heterogeneity in the tumors? And then again, we went back to the naturalist approach, basically the same approach that we're using to measure biodiversity in an ecosystem can be used to measure diversity in a tumor, which is basically counting the number of species that are present in a particular area and also counting the individuals within each species. So we decided to use this approach, and in our case, a species is basically a unique cancer cell that we identified in this case as a cancer cell with a particular copy number for a particular locus. And then we counted how many cancer cells we see with that particular phenotype in the CD24-44 population, in the DCIS, and in the invasive areas. And this particular plot is basically depicting the population diversity from the point of view. This is looking at the histogram of species or cancer cell abundance. The x-axis shows the copy number ratio that we're counting, and the y-axis shows the number of cells that have that particular copy number ratio. So what you see that there are tumors where the different populations seems to be actually fairly similar to each other within the tumor, the DCIS, invasive area, and the two cell populations as well. But there are very dramatic examples where the CD44 plus population is much more diverged than the 24 plus cells, and also they change as the tumor becomes invasive because the invasive area, again, is very different. This is another example and another example. Then we actually, another way to calculate this or compare this diversity among tumors is to calculating the Simpson index or the Shannon index, which is basically ranking the diversity of the species to measure richness and evenness, which is basically like how many species again and how many different numbers each one has. What this shows here, that again, like the 4 different cell populations in the same tumor, most of the cases seem to have the same or similar diversity, but there are examples when, for example, the invasive area has lower diversity than the DCIS area. And overall, as you see, like the basal-like tumor seems to have higher diversity than luminal tumors, at least based even this limited subset. So we basically think that this kind of approach is a very useful way of looking at the tumors. Unfortunately, it's a very labor-intensive way because we have to count single tumor cells in the tumor, so it's a lot of work to do these studies. And this particular slide I really like. It was from a review by Carlo Mello, who's been one of the pioneers of this ecosystem approach to cancer. And basically, it depicts like what is the rationale behind this very high degree of diversity and how can diversity actually change with time. So if you imagine in a solid tumor, the different parts of the tumor actually have very different environments, meaning like the periphery of the tumor, the middle of the tumor, exposed to very different conditions. And therefore, the cancer cells that are most able to grow are very different in the periphery and in the middle because they are exposed and selected to different conditions. As a consequence of this, if you're looking at the tumor as a whole, the diversity is continuously increasing as the tumor is growing because each area evolves on its own. Occasionally, particularly, for example, after treatment, when you're killing cancer cell with a particular property, you can actually decrease diversity because the resistant clone can predominate, but then eventually these will further evolve. And basically, based on a diversity score, you can actually kind of determine the evolutionary history of the cancer and how long it took to grow to that stage. So anyway, we're looking at this at more detail in patients before and after treatment and different stages of progression using these same approaches. In addition to the genetic heterogeneity, I mentioned that we also study epigenetic diversity, such as DNA methylation patterns. This study we published last year, basically, we looked at the global DNA methylation profiles of the CD44 plus, 24 plus cells from normal tissue samples and also from breast tumors. And what we found that they have very unique DNA methylation patterns. These diagrams here shows the genome from chromosome 1 through Y, and basically the y-axis shows the ratio of the genomic spots that seems to be statistically significantly differentially methylated. What you see here that the majority of the spots seems to be higher in the 44 plus cells, which basically using the approach we studied this methylation-specific digital karyotyping suggests that those cells are hypomethylated overall compared to the more differentiated cells. Just to look at this a different pattern, we actually aligned all the samples that we studied based on a hypomethylation score. And again, what you see that this 44 plus cells seems to be more hypomethylated than the CD24 plus more differentiated cells. This actually gave us the idea then, can we actually convert the CD24 plus more differentiated cell to a more progenitor-looking 44 plus cells by basically making them hypomethylated, which can be achieved by treating the cells with azacitidine. So in this experiment, we treated the cells. These are epithelial CD24 plus cells from a particular tumor, treated with azacitidine. They underwent a very dramatic morphologic change in a few days. they lost epithelial morphology, and they also became more migratory and more invasive following azacitidine treatment. And they also started to re-express genes such as TGF-beta type 2 receptor that was silenced in the more differentiated cells due to methylation. So what this shows that basically by changing the epigenetic pattern, the global epigenetic pattern of the cells, you can actually acquire this stem cell-looking cancer cell phenotype. Then when we looked at the actual specific genes that seems to be differentially methylated between the more differentiated and less differentiated cells, we found that a high number of genes that known to play a role in development and stem cell function, such as homeobox genes or TCFs or SMEDs, they seem to be hypomethylated and expressed in the progenitor-looking 44+ cells and silenced and methylated in a 24+ differentiated cells. So again, here what we thought that maybe one or a combination of these transcription factors could be actually responsible for this phenotype, and therefore if we would express this transcription factor in a more differentiated cells, then we can actually reprogram the cell again to the stem cell looking cells. So in this particular example, we overexpressed FOXC1, which is one of these transcription factors we found differentially methylated. We expressed it in this epithelial-looking MCF12A cells, which has nice equatorial localized to the membrane. Following FOXC1 overexpression, the cells completely lost this epithelial morphology, became more mesenchymal-looking, and again, they became more migratory invasive, and they started to express the genes that are more characteristic of the 44+ cells. So in this particular example, what this shows that in a particular cell type, this doesn't happen in every cell. So as Bob said, there is some inherent property of the epithelial-looking cells that can determine if they behave this way. But basically, just the expression of one transcription factor is able to switch the phenotype of these cells. And lastly, I'd like to close with the most important question is like, what is the clinical relevance of this heterogeneity? I already mentioned that this intertumoral heterogeneity is very important for personalized treatment because basically every tumor subtype seems to behave like a different disease, and in terms of like require different treatment, and also the prevention of the different disease likely to be different. But this intratumoral heterogeneity is actually poses a major problem in terms of like therapeutic resistance, and also diversity is likely to contribute to progression of the disease, just based on the fact that the higher number of different cancer cells or species you have, the more likely to have one that is able to resist the treatment and also progress. So one of the approaches we use to look at this clinical relevance is to look at the signaling pathways that seem to be differentially activated between the 44+, 24+ cells. And this just lists some of these pathways that includes TGF-beta, hedgehog, and many others. And this particular pathway is the reason why we were interested in these, because there are many therapeutic approaches currently designed to target these particular pathways with the hope that that will be more effective way of treating cancer than using chemotherapy. So when we tested actually one of these inhibitors, this particular case looking at a TGF-beta receptor kinase inhibitor, how the cells within the same tumor, but different phenotype would respond to this treatment, what we found that these 44+ cells, which as I mentioned have activation of this pathway, they actually undergo very dramatic response in terms of morphological changes following TGF-beta receptor inhibitor treatment. They have abnormal beta-catenin, equatorial, and ZO1 before treatment. After treatment became very nicely epithelial looking and membrane localized. But the 24+ cells in the same tumor did not respond because they don't have this pathway activated. So that just shows this complexity again, that if you're trying to target a particular pathway and it's not active in all of the cancer cells, then it's not going to be an effective way of treating the tumor. Another way to look at the clinical relevance is to see whether the clinical signature, the gene signature of the cells that differentiates the more stem cell looking and differentiated cells correlates with clinical outcome. So what we show here that the patients who have tumors that have high expression of these stem cell looking or CD44+ genes, they seem to have a shortened distant metastasis-free survival, which is consistent with the model that these cells are most likely to migrate and invade. But then when we looked at the distant metastasis of the same patients, in this case, we had the matched primary tumor and the distant mets, and just looking at the 44-24 staining pattern, what we saw, to somewhat the surprise, that they seemed to be more CD24 positive cells in the distant mets, and this was actually true in 14 out of 14 patients, and regardless of the site of the distant mets. And 44 staining pattern was not really consistent, and it didn't show this difference. So what this suggests, as have been implied in the talks before me, is that maybe there is a transient phenotype conversion to these more stem cell-looking CD44 plus cells, and those are the ones that initiate the metastasis, but then they can switch phenotypes. Or maybe we were actually wrong, and these cells can acquire property to migrate and invade with some other mechanism. But just to be sure, it's not just CD24 expression that's different. We also confirmed by DNA methylation patterns that actually the metastatic sites seems to have a methylation profile that suggests the high number of CD24 plus cells, because we picked 4 genes, 4 transcription factors that were more methylated in a 24 plus cells, and the metastasis consistently showed higher methylation for these. So that again shows that a metastatic site is a different site from the primary, and if you combine this kind of genetic and phenotypic heterogeneity with the genetic heterogeneity that I've shown, that we actually seem to have a very fairly daunting problem in terms of trying to treat these patients with a single or even a limited combination of treatments. So basically our conclusion is that these cancer cell phenotypes are determined by the combination of genetic and epigenetic alterations in the tumor cells, and they are influenced by the microenvironment. These phenotypes can be stem cell looking and more differentiated phenotypes, but these can change, and basically like a tumor evolution is similar to the natural evolution in the ecosystems. And clinically, it's a very important problem, and we think that the approaches that we're using, looking at population of cancer cells instead of just the mass, will be more informative and hopefully will be more helping to overcome the therapeutic resistance and better treatment. So with that, my time is up. But I'd just like to acknowledge the people who did the work. In my lab, Mikhail Shipisky and Lauren Campbell has worked on the cancer project on characterizing the 44 plus cells. Soyun Park, Hee-Jun Kim, and Vanessa Almendro has done these very labor-intensive but beautiful immunofish studies. Noga Kimron did all the normal work, and we have numerous collaborators, clinicians, and pathologists, different institutions who are helping up with this issue. So, thank you.